Life runs according to a blueprint, which is the DNA. So we can refer to the DNA as life's own sequence. Now, what is DNA? First, we all have proteins and enzymes in our body. These enzymes and proteins are created from different processes, all of which are coordinated by the DNA. DNA is the reason why one person is different from another. Now that we understand what DNA is, we can talk about the gene. Your gene is made from the DNA in the form of codes. These codes usually take the form of a four-letter combination containing alphabets of chemical basis, i.e., the ATGC, which is also called the nucleotides. If the cell is ready to create any protein molecule, it takes instruction from the gene sequence. Thus, the gene sequence determines the type of protein molecule that will be created, and therefore, the proteins are unique eventually, especially in shapes and functions. The uniqueness means they can control various segments of metabolism, cellular anatomy, and physiology. Another important component to discuss is the genome. In simple words, all the genes found in the body of any living thing is collectively referred to as a genome. For humans, our genome comprises about 20,000 genes. Genome, however, differs in composition, another reason for our uniqueness as humans. And this leads to differences in gene sequence, which ultimately results in variations in function, structure, and the amount of protein produced. Now, let us compare mutation and polymorphism. Mutation is the consequence of rare differences in the gene sequence. When this happens, its effect on the population is negligible, and, most times, we can predict the outcome. Polymorphism, on the other hand, is more rampant, but with less serious effects relative to mutation. Also, it is usually influenced by some environmental factors. You can have polymorphism in different forms, but we will be discussing only the most common type today, which is the single nucleotide polymorphism, or SNP. SNP simply means the swapping of one nucleotide for another. Note that the genome contains up to 10 million SNPs. You will find people use SNP and mutation interchangeably. However, an important point about SNPs is that they rarely affect the protein created, and in those rare cases, they are not of medical importance. One of the most notable effects of SNPs is the distortion in the nutritional requirements. This causes the body to respond to most lifestyle and dietary factors differently, and this is where genetic testing comes in. With genetic testing, it is possible to get vital clues that could help in coming up with a bespoke special diet and protocols for each person. Although nutritional genomics as a field has continued to evolve, there is still more to do as per the exploration of SNPs. In-depth investigation of these phenomena will reveal the beneficial forms and how they can be applied clinically to the interpretation of the genetic test information. A couple of factors influence the final interpretation of any SNP on the psychology of an individual. These factors usually include the diet, environment, as well as the epigenetic and genetic factors. The field of genomics, if explored well, is capable of making common SNPs very relevant clinically, especially when it comes to nutrition. From our discussion so far, it is clear that the DNA has so much to do in the future of human health and nutrition. Let us start with the Integrative Nutritional Genetic Analysis, Inga test, which was inspired by the need to understand DNA with regards to human health and nutrition. With the Inga test, it is possible to find out the most beneficial enzymes in your body and how to best use them to improve your health. By identifying the different SNPs in your gene, all dietary inconsistencies, if any, will be revealed. Also, if you have an insufficient amount of nutrients or antioxidants or none at all, this test will also make this apparent. The dietary inconsistencies, if any, can be easily solved by creating a personalized nutrition protocol for the patient based on their genetics. With this protocol, the patient gets vital supplements and nutrients that can make up for losses and neutralize the inconsistencies. They also help to ensure the judicious use of vital enzymes, as well as the creation and support of healthy cells. If need be, The patient may also undergo detoxification therapies. This is not as complex as it sounds, especially when you consider that our well-trained nutritional consultants will be with you at every step of the way. And by towing the path of nutritional support, we will arrive at a viable maintenance protocol in the end, while providing lasting solutions to all issues. The patient also gets a bespoke maintenance program that meets all their nutritional health requirements. The more we dive into this progressive approach of providing excellent help and support to genetic variants, the more we come up with excellent solutions and answers to several challenges. So, why not join us today?